começar? Eu não sei o nome dessa ciência.
Que na verdade é o que linka os nossos três papéis.
So, good morning, everyone. So we begin the last session of this morning. I'm Flavia. I'm postdoc researcher in Siddhartha Lab, and this session is called something like solving visual problem, and binding problem. And we have three professors from our house. The first speaker is Sergio Neuenschwander, and he will present entrainment in dynamics in early visual processing. So thank you.
Thanks a lot. Thanks a lot. So, our lab has, in fact, two presentations. This is mine, and then there is, in the next day, another presentation. And this will talk about mainly our work in cats, the entrainment dynamics in the early visual system. Central dogma in neuroscience, particularly in vision neuroscience, is of course the neuronal activity encodes attributes of the physical world. So this is a nice section of the retina, light cross, the whole retina, activates the photoreceptors, and you have as an output the activity of retinal ganglion cells. So this very basic idea is nicely captured on this data from '96 of Christian and Ruth Kugel, where you see the responses of a non-X cell of the retina of an anesthetized cat. And here you see, you have an idea of what will be the perception of a stimulus that consists in varying the spatial frequency as a function of contrast. And this became a dogma. And I try here as an example of how powerful this paradigm is. In some recordings we made in a patient in Goiás, now 1.5 years ago, and we could record from the lateral geniculate using the very same, this is important to say, techniques as we use in the past in cats, mice, and monkeys. The reason for doing this type of surgery is to treat focal epilepsy. So essentially, the patient goes to a surgery where electrodes are placed in the lateral geniculate nucleus, and then we are able to record the spiking activity of the neurons. As I said, exactly as we have done or we do with our animal models. The data we could obtain in this single session allowed us first to map the receptive field using a moving bar. You see here the modeling of this receptive field using a difference of 2 Gaussians, very nice data, in fact, because it was a very short period we had available for doing the experiment. This, important to say, is the first time we obtain spiking activity from the LGN of a human being. And from this notion that if we could describe the properties of every receptive field, what we simply say receptive field properties, we could build the big picture. That's why I told Flavia to say that we have a kind of a solving the visual problem, because this is always an important and difficult question in vision science. And some results from this surgery. So here a little bit of more detail, where you could connect the microelectrodes with this system. And there is no activity. We started beginning the recordings, and we prepared some stimulation paradigms. We, of course, started then looking for receptive fields, exactly as we do in our animal experiments. And we have, in fact, a very interesting paradigm also including attention, but you see how nice are the spiking activity you could obtain in these conditions from the human brain. Testing figure ground segregation. And again, I hope you see nice spikes, activities that can be then sorted, and you obtain then these response histograms. So this is a premiere in the sense that nobody has ever obtained something that, of course, we expected from the physiology we have done in monkeys, but it's very nice to see this in a human being. You see nice red-green opponency of this cell. So we have different properties of the stimulus that could be green, red, blue, and yellow. And you see that particularly this unit 10, they show a very clear red-green opponency. This unit 11 has only a very clear selective response to the red stimulus. So this is the basic thing. You can obtain response and count spikes and measure the selectivity of these responses to certain aspects or features of the stimuli. Nicely also you see difference of the two channels, the Magno and the Pavo channels or system. You see here in this patient 2, the response for stimuli with different spatial frequency. You see that there is a very clear tuning where the cells preferred the lower spatial frequencies, here unit 13, and also we measure the temporal frequency. So a basic characterization of the response, and you could, of course, replicate what we knew from our cat and monkey experiments. And I brought this example of Christian and Ruth Kugel from 1906. You see, he proposed first the existence of two independent channels in the retina, the X and Y cells is exactly what you see in the human brain 60 years after. So from these, now one paradigm is characterizing the response of the cells and trying to understand how they represent certain features. But of course, the problem is more difficult because we need also to explain how these features are combined into whole objects or whole independent objects from the background, like this example. Claudio will explore also this. Then the term visual binding. One possibility is the use, that the brain use oscillations as a way of building these relationships in the temporal domain. So this is a kind of extension of the first paradigm where only the single cell responses could explain perception. People start exploring the fact that there is a lot of oscillations, and we'll see many examples from our own work here in the Institute that oscillations are very prominent. And just a question, because I'm a little bit lost with the timing. Now it's 11:25 when I'm supposed to stop, because we started very late. In 10 minutes. Mm-hmm. I have 10, only 10. When I'm supposed to stop. Now is 11:25. 14. So here is an example of strong oscillations in the retina where you see a link with this binding problem. Very interesting study made in vitro from the group of Bloomfield in the Sun University in 2017. You see that when you show in vitro, so under the microscope, a stimulus that consists of two patches, if you now make this stimulus collapse into a single one, there is the appearance of very strong synchronous oscillation. And they show that this depends on the connectivity of the amacrine cells, because in the retina you have a lot of horizontal connectivity that could explain this horizontal integration at the level of the inner plexiform layer. And we characterize these oscillations in the cat, and the first thing we show is that it depends on the intensity and the size of the stimulus and the continuity. So this became very interesting because it could be that oscillations already at the level of the retina could contribute to binding by phase locking of oscillatory response. This was the very idea. But for our surprise, these oscillatory responses show to be very dependent on the levels of the anesthesia. So this was kind of unexpected. You see in this graph that the coherence decreases if the level of the halothane inhalatory anesthetics decreases. So more anesthesia, more oscillations in the retina. Then we started to record also from our cats and also under other conditions, other anesthetics like ketamine. And you see here a clear difference. These oscillations that were so prominent under halothane were absent in the awake state. This was a kind of a shocking result for us. And I was also very happy, and you see how fruitful was this experiment, or single recording sessions from a human being. I have no time to explain, but you see that when you search for gamma oscillations in the LGN of awake, because the patient, I have to mention, was always awake during the surgery, you see no trace of oscillations, just reinforcing our data obtained in the cat. Now, if you record from the LGN of a awake cat and you show your stimuli in a CRT that characteristically has a flicker, interestingly, the response of the cells in many times are able to follow these very fast rhythms. This is very interesting because it shows how temporally precise is the responses in the early visual system. In fact, this is almost amazing. We could see responses at 120 hertz as a limit that we could measure in the awake cat. And we started then comparing two stimuli conditions, one with LED, where you could then have a completely flat stimulus, so no flickering. And you see, in the case of the awake cat, oscillations that we call now, and you should mark this term, entrainment, because this oscillatory input entrain the activity of the cells, appears only for flickering stimuli. If you show LED stimuli, or a stimuli that was produced by a LED that is flat, there is no oscillation. So here the power spectrum of the autocorrelation are completely flat. Now, why this is interesting? It's interesting because it shows first how precise the visual system can be, and second, it raises the possibility that the oscillations we have seen under the anesthesia are simply due to synchronization mechanisms in the retina that are in place, of course, when the cat is awake. So oscillation is a kind of special phenomenon that arise from perturbation that the anesthetics does in the retina dynamics. However, the synchronization should be there. So first I show some examples here using first entrainment with the monitor at different frequencies, 75, 120 hertz. You see very clearly that the cells are capable of following this fast oscillatory input, but also for lower frequencies, 60 hertz. Whenever you deliver ICe-UFRN or halothane, and then these different stimuli that was produced at the different frequencies collapse into a single limit cycle oscillation in the retina. Why this is interesting? As I said, this is interesting because because it shows up the synchronization phenomena. And quickly to show two studies, this from Alonso, demonstrating that synchronized spikes are important for eliciting spikes in the cortex. So from the LGN to the cortex, if you have synchronous spikes, this increase the likelihood of having second spike in the cortex. And also the very important nice study from Marcus Meister, where he says, analysis of receptor field showed that concerted spikes encoded information. So from the beginning, when I said that one paradigm was the encoding based on rate levels, you see the proposal here already at the level of the retina is that the concerted, so the synchronous firing, could encode properties of the stimulus. A recent study has shown from Stock in Neuron in 2017, I cannot go into detail, but has shown that this oscillatory response in the LGN comes from the retina, exactly as we have demonstrated in our cats experiments, so here in the mice. But if you see carefully here the data, you see that in the histograms that these oscillations are synchronous. And this happens likely because they are entrained. I have even written to Starkey, and I had not a clear response because I said to him, how is possible you see these very nice synchronous response in the histogram if they are not entrained? How this would be possible? He gave me a not very clear response, but it became clear for me that the entrainment is a very common phenomenon in the visual system, not only of the cat, but of the monkey. We could obtain the evidence for this in the visual cortex, and of course, in our experimental model, the cat. So we then start exploring exactly this, studying the entrainment of the responses at the level of the LGN and the retina. And you have some predictions based on our previous studies in the cat. And one very important is that the entrainment should also be dependent on space continuity, the size and the intensity of the stimulus. At the end is like this. What we have made in the past using the anesthesia does not show exactly or does not explain or does not help to explain the binding problem. However, reveal some synchronizing properties of the retina that could be also be captured with a simple paradigm as a flickering stimulus. So we decided to develop a new stimulation system that consisted on a tripartite circles, so a series of concentric circles, and we could then, in addition to the monitor, we could then drive LEDs from different families at different frequencies. This is very interesting because now you can impose conditions where you have non-continuous flickering stimuli, conditions where you have pieces of the stimulus made of different frequencies. And you see how these drive the network at different frequencies and how they interact at the level of the retina. So here, to show quickly how these are made, these are concentric cycles. And we could, using, of course, my wonderful skills in LabVIEW, do different paradigms where you could have frequencies different in 1-2 hertz. But there are other things you can do with a LED that are wonderful. And one is if you have a square waveform, you can change the phase and the duty cycle of the stimulus, things that are very difficult, if not to say impossible to do a monitor. So we start recording from the neurons. Before we characterize the properties of the cells in the same way we have done in the human algae, and mapping the receptive fields and then classifying the cells. We use also, or trying to use these reverse correlation techniques, and use different, now I have a battery of different anesthetics. That is also very interesting to see the effects of different anesthetics in the entrainment dynamics in the retina. So use up to now halothane isoflurane that behave in the same way, and ketamine and propofol that behave also in the same way. There is a big difference here that the halothane and isoflurane induce intrinsic rhythms in the retina, and the ketamine and propofol not. So you are more easy to see entrainment with ketamine and propofol, different from isoflurane and halothane, where you always, the retina fall in these intrinsic limit cycle oscillations. And then we characterize these flickering stimuli, changing systematically the frequency, intensity, spatial temporal continuity, and we have done other type of experiments that I can explain if someone want to. So here, only to have an idea of how these experiments are made. We start first characterizing the responses with bar. We acquired the data and to compute the differences in shape of the spike waveforms for spike sorting. And then we can also use this kind of noise stimulus in order to obtain the reverse correlation maps of the receptive fields. So you see here that this cell respond mainly for the dark dots, so this is an off cell. Now the results, briefly, first we confirm our hypothesis. If there is a gap in the flickering stimulus and then the entrainment is gone, if you show with low intensity only the center, the cell cannot follow very fast flickering input, so there is a limit up to the cell can follow. And now if you have a big continuous stimulus, and then there is a very strong entrainment. So somehow this replicates exactly the results we, Starkey and Bloomfield had in the algae and in the retina. Explaining maybe why these oscillations are correlated in these conditions under anesthesia by halothane. Now, if you show just the center, so the stimulus that is flickering is just the center, with comparison now with low intensity and high intensity, this should be 50 candelas per square meter. And you see that if the stimulus has low intensity, the cell stops following the flickering of the center only up to 20 hertz, so he stops. However, if the stimulus has a very high intensity, and then the cell can follow it in fact, up to 100 to 120 hertz. So here I show data up to 60 hertz. This is very interesting Because if, and this has been characterized in the past, here the work of Lem-Cooley in 1980, the cells, they have a cutoff for the temporal frequency. So we show here that this cutoff depends on how the cell is, so to say, synchronized with other neurons. And this is revealed by these flickering stimuli. So here you see this part of the data. I have not an explanation till now, but I show because it's so interesting to have a decay. I needed to check this. But the cell here is clearly following up to 90 hertz. In this case, with a stimulus that flickers only in the center, high intensity. Now, and I show two more experiments in order to finish. This is so interesting because now what we change and introducing, so to say, discontinuity in the stimulus was not appearing or not the stimulus, but what we changed was the phase rotation of the stimulus. So if you see this stimulus, you think nothing is happening there compared to the one that has no phase rotation. If it is, for instance, at 60 hertz. In this case, it was exactly at 60 hertz, 63 hertz. Now, what happens here? This part of the stimulus is phase rotated in relation to the center. So although you do not see that is phase rotated, the cells are able to detect this, if you wanted to say. This is, in fact, a very poor description of what I'm saying. The cells are sensitive to phase rotation in the flickering stimuli, showing that, in fact, the retina, all this timing is extremely relevant, and the cells are very sensitive to these very fast frequencies. And here, another example that in a single recording from two cells from the same electrodes, that, so to say, summarize our results. If the stimulus is continuous and flickers here about 100 hertz, the cells can follow. If the stimulus is large enough to recruit a very large population in the retina. If you introduce a discontinuity and then this entrainment disappears. If you introduce phase rotation in parts of the stimulus, this entrainment disappears, and also as a control, we show only the periphery, this disappears. We wanted to explore these data and these results in two contexts. One, in our new set of, or a new research line we want to open in the lab related to the Alzheimer's disease, because it has been now shown that flickering stimuli can entrain pieces of cortex, the visual cortex, maybe the hippocampus. There is a lot of controversies still on this. The group of Buzak is saying that they do not propagate in the brain. However, it seems that this may be a promising treatment for Alzheimer's disease because it improves the clearance of the beta-amyloid plaques. So we think that this could be very relevant if we understand from the retina how this entrainment dynamics happens using this different kind of stimuli. And I wanted to acknowledge my collaborators. So this work in humans was made with Matt Salve, Peter Hursma, and the wonderful group of Osvaldo Vilela, that is the neurosurgeon in the University of Goiás. Peter comes from the Amsterdam and the University of Amsterdam. And also the previous work in the retina was made with Natalia, Giovanni, and Sexta-feira, Fabio Freitag. And mainly I would like to invite you to see the poster of Lucas. Hello, Lucas. And the presentation of Estevam, that he'll be talking about other things, but also very interesting, next day. So thank you.
So thank you, Professor Sergio. The session is a little bit late, so my proposal is that we have the questions on the end of the session. It's okay? Or we have time for one question, Professor?
Yes,
yeah.
You prepared a question for me.
So, one question, or we can follow? Okay, so in the end of the session, we have some minutes to make questions. So the next speaker is Professor Kerstin, and the lecture is from interhemispheric integration to visual perception.
Thank you, Flavia. Thank you, Sergio, for this very interesting talk. And I prepared something to introduce a little bit the trajectory of my lab, coming from postnatal visual development, studying intercortical connections, clustered intracortical connections in the visual cortex in animal models, and then how we come to study visual perception nowadays in my lab. So from animal experiment to human perception, so one thing I want to mention is we studied here cortical layouts of orientation preference maps in our lab. So why are there different layouts actually to represent visual features in visual cortices? So we are now coming from the retina to the algion. We are now coming to the primary visual cortex. And do these different layouts maybe have perceptual strategies which are different? And then another question we ask in my lab is, do selective cortical connections implement these perceptual grouping strategies? So if there are different layouts, maybe there are different strategies. And could ongoing activity patterns, which are rooted within the selective connectivity, reflect prediction of shape and motion in vision? And we could call these patterns maybe priors of what there is to see in our visual environment. And from that on, we part to the question, can we actually define behavioral signatures of selective interhemispheric connectivity in human observers? So most of the work was done about the interhemispheric connections, and I also explain you why we are getting from the intrinsic circuits to the interhemispheric connections. So we start in the retina with Sergio and went from the corpus, from the lateral geniculate nucleus. Now we are going to the cortex, and this is just to show you, of course, there are several visual areas in primates, so in us human observers dealing with vision, and basically what happens after the geniculate nucleus, and this happens already in the retina, as you have seen in this very nice talk, is that we have representations of visual features in the outside world, and the big question in visual neuroscience is how we link them together. And the biggest comprehensive representation we have in the primary visual cortex, and from there on we have topographically organized cortical visual areas which partially process these features in kind of a hierarchy, but we have also a lot of parallel processing. We have cortical connections within the cortices which can already link features from outside the classical receptive fields already in V1, and that's where I'm staying. And then how do we come from V1 to the human observer? So just to remind you, so you learned a little bit about these maps already in Sigrid's talk. So most of the animals we studied so far, they have this nice periodic orientation preference map where this easy contours, so now I just walk around, where this easy first order contours, I call them, like stripes, contours of a certain orientation. You see that in the retina and geniculate nucleus, the stimuli were circular. Now we are going a step further, we go to primary visual cortex, and now the stimuli which the neurons prefer, they are like stripes of different orientations, and the neurons in the primary visual cortex of This is cat, and also in primates and in humans. This has to be, it was shown also with functional magnetic resonance imaging, that these kind of structures exist also in human visual cortices. We have this periodic maps where neurons representing these first order contours repeat in circles, and then they cluster around singularities in an orderly manner. And this is actually, as I told you, so this happens through very mammals, a lot of mammals, so all primates, and as carnivores, and interestingly, not in the rodents. And one question which rose when I was still in Germany, actually, with Fred Wolf, who is here today, was, is this actually this pinwheel-like nice layout of orientation preference maps? First, is it important to actually represent visual features? So does it has an impact in vision? And the second question, is this actually the fact that we do not see it in the little animals, which are so common in neuroscience, not only in visual neuroscience nowadays, in the rodents, has it something to do with the size of the visual cortex? And here in the lab, we could we had access in the northeast of Brazil to a large rodent with a very large primary visual cortex, which is the aguchi. And the group I had at that time made the experiment happen, which was a very efficient group around Dado Ferreiro, Sergio Neuenschwander, and João Patriota at that time. And surprise, surprise, the aguchi doesn't have this pinwheel-like layout. It doesn't have the typical periodic maps where orientation preference clusters gradually around these pinwheel-like structures. And what you can see already here, these are optical imaging maps of intrinsic signals. So the darker it is here, so if we go back, so I switched a little bit here. If you see here, neurons in a cat responding to vertical contours, and here responding to horizontal contours, they make complementary maps. They make these stripes and beaded bands, which we all know from the literature. And now you go into the aguchi, which has a cortex is almost the same size of a cat. You see, it's a very homogeneous map. So all over the place, apparently, we find neurons which respond to contours of horizontal stripes, and to vertical and to oblique stripes. But what struck me at that time was that the map for the horizontal contours is much more dominant. So there seems to be not only the lack of the layout, but also there is a bias, and this is the horizontal bias, which was also confirmed in the electrophysiology. So apparently the aguchi and many rodents and lagomorphs have this too, have a bias for horizontal orientation, which might have to do something with their retina and their horizontal streak. They have a different retinal layout than the humans and the frontal-eyed animals. And humans, on the other hand, have a different bias. And not only humans, this cardinal bias was shown for many frontal-eyed mammals, also for cats and ferrets. And I show here only some psychophysical behavioral example. So it was both shown in the physiology and also in the behavior of the humans that they have a cardinal bias, cardinal for both vertical and horizontal. So we as human observers are actually better to discriminate little orientations. So Sigrid showed the experiment of the mice who were having to discriminate orientations, and I should have asked that question at that point. And now take us humans and look at this famous Poggendorf illusion, where the interruption of the contour induces an illusory break, which isn't there because the contour is actually continuous. It's just behind the mask. And I don't know if the effect gets through the audience, but if the line behind the mask is oblique, the illusion is much stronger. So we are much more sensitive if the lines are vertical. The same for the herring illusion. This was quantified by Gerald Westheimer as well. So the amount of curvature we have to, so with this, you all know this illusion, these lines don't look straight, they actually look a little bit curved. And the amount of curve you have to put to compensate for this illusory curve is actually higher in the oblique cases than it is in the cardinal cases. And this is a behavioral reflection of the fact that we are actually very good at vertical and horizontal, and a little bit less good in oblique. So is this something which we can make use of? Is this something useful? And do orientation maps actually reflect this? Yeah, they reflect this, and there's a very old finding already, like 20 years ago, more than 20 years ago, of Amos Ariely's lab, that actually this periodic pinwheel maps, they, and we reproduced this finding here when I came to Brazil with Thiago Altavini and the group which was still coming over from Germany, is that ongoing maps of this pinwheel-like layouts, they pass spontaneously through the maps which look like the evoked ones, and they go through all orientation selectivities, but when you do a quantification, you actually see that the maps preferring 0 and 90, which is horizontal and vertical, they appear much more often spontaneously. So the cortex is not visually stimulated. It somehow goes spontaneously through the network. Something in the network gives this bias and lets these spontaneous maps appear more often. So from that now, we have to come to the human. Do we actually need these periodic maps to perceive form and motion? And this is a question I will not be able to answer, but then we have to do some behavior, and it's an interesting question. But we could ask, how does perceptual grouping actually operate? And this goes back to my thesis. And you see that perception actually uses criteria to group contours, because now we have to go all the way from the primary visual cortex to behavior. So the question is, which contours are in this image? So if I go back, you might see a lots of Gabor patches which have different orientations here, like in the J. Borges xylografias. Yeah? So bigger elements are made out of smaller elements which have the same features, and these features, according to the traditional old Gestalt psychologists, can be proximity. So if contours are next to each other, if they have a similar shape, or if they have a similar color, like in the picture, in the xylogravure, or if they are parallel, or if they are collinear. So if you would, you saw it already, some of the lines here pop out maybe, and why do they pop out? Somebody sees other lines here in the picture? Yeah, a lot of, yeah, so there are others here, yeah, and they pop out a little bit because, so this is called collinear facilitation, because not only have they the same orientation, but also they are aligned along a collinear line. So two Gestalt criteria actually in place here, apparently making this facilitation for the human observer. Now we're going back to my thesis, and Secret has to laugh. So the primary visual cortex actually already has a network for this, which can implement this perceptual grouping criteria. And I think it's already like 50 years ago, there was found, so most of the time people were only talking about the networks going into the cortex and the columns and the vertical structures, but then at some point people became aware of a dense network, primarily in the supragranular layers, but also in the infragranular layers, where neurons are linked to each other, and it's not that neurons just randomly connect everybody which is in their environment, but they do this clustered intrinsic connections, that's what they are called. And if you, so you see here a neuron, you see it branches its axon parallel to the cortical surface, and it makes boson, connects to other neurons, and you see it doesn't connect to everywhere, it's quite selective. And when it was possible technically to superimpose those networks to the colorful orientation preference map, it became also clear that the two things have something to do with each other, that actually the neurons which are interconnected here in the colorful map, and this is not only in the cat, and here's the tree shrew, it is also in primates, actually in all maps, in all animals which have the modular maps and were studied in this direction, you find that if you make an injection here, for example, in the green domain, which prefers oblique contours, you find that with very high likelihood, other neurons in other green domains farther away and having receptive fields somewhere in the visual field are interconnected to this neuron here. And the network is reciprocally, this Kishwader and Eisel showed. And we showed it in my thesis with Secret also in the retrograde direction. And not only what you see already here is, and I designed it here so that it actually pops out, it's not that everybody in a circular radius is interconnected, it's making an ellipse. And this ellipse is not random with respect to the orientation preference which is studied. So it turns out if we have neurons which were actually injected into a domain preferring horizontal contours, then the network, the ellipse of the network, not only connects neurons preferring horizontal, but also neurons which are along a line projected, represented in V1, collinear to the shape which is actually here stimulated. So two perceptual grouping criteria represented in V1. Now I told you I'm coming to the interhemispheric connection. So this network, it doesn't stop actually here at the midline. So as you who studied sensory systems in some of our classes know, the environmental periphery, so our sensory periphery, not only for vision, but also for the body perception, also for hearing, for olfaction, I don't know, is actually split in two halves. So this thing having two vases, and who actually makes the symmetry in our brain and restores the image at the vertical midline, which is actually split into halves. So this vase is represented in your right brain, and this vase is represented in your left brain, but you don't see actually that you are looking at this image with true brain halves and that they ensure the continuity at the midline. But this connection is very important because it's active actually all the time. Important for the next part of the talk is actually that the connections I was talking about, they're not only selective for orientation and collinearity within one hemisphere, but also if we go to the other hemisphere. In fact, if we are very, very close to the vertical midline of the visual field, the ellipse is cut in half because we are so close to the vertical meridian, and the network then continues on the other side. This was something we saw only years later when we looked at the injections we did at that time, but we didn't have enough data to quantify it. But unfortunately, other people did the experiment to show that callosal axons going through the corpus callosum to the other primary visual cortex actually adopt the same features. They are selective for the same contour. So if you have a neuron here on this side of the brain which prefers vertical, the likelihood that another neuron on the other side of the brain, which is interconnected, prefers also vertical is very high. Yeah? It's not 100%, and there's a big polemics about how selective really these connections are and how 100%, nothing is never 100%, but it's a bias. And we started to study these connections functionally as well, and you know this from talks from maybe for years and years and talks I gave here in the Institute. We deactivated one visual cortex and looked what the other visual cortex does in the absence of this input which comes through the corpus callosum, and there we could actually also demonstrate functionally that there's a selectivity for the same orientation and for collinearity. So here an example of receptive fields very close to the visual midline. You cool the other cortex, which is supposed to give via this selective connection input to the neuron in V1, you see that this receptive field shrinks, but not all receptive fields shrinks. They shrink in a selective manner. You see that this one doesn't, and this has to do with the preference direction. So we saw here in this experiment, and actually here is the same finding, here is the same neuron actually. If you stimulate the direction which comes out of the cooled hemifield, then the activity drops in kind of an expectation of a stimulus which is not coming. Yeah? And if the stimulus goes into the cooled hemifield, there's no such an effect. So only when there's really a direction-selective stimulus coming and crossing out of this cooled hemifield, which is supposed to give some kind of prior input to the visual cortex, is coming out of this visual field, then we see this drop. But this was visually activated. So I showed you these maps, which are there spontaneously all the time. I, somewhere in the talk, got lost talk about priors. So if there's some kind of, maybe these networks do some kind of prediction, they help to implement the grouping criteria by facilitation. And I'm talking here about prediction, so then we actually have to take away the visual stimulus. If we did this in this very first experiments with the voltage-sensitive dye imaging, and what you see here is actually here frames of these periodic maps recorded with voltage-sensitive dyes in a cat, yeah. And here you see the activity in the baseline, which is, so there's no cooling going on, no deactivation of callosal input. You see here that in the baseline, we see the same effect like Kenneth and Ariely. So we have, usually the network goes through maps and it goes spontaneously through vertical and horizontal with a higher likelihood than it does go through other orientations, as if it would be expecting all the time some kind of symmetric priors. And if we take away the input from the other side, you see that the color distribution in these frames, so every frame gets a color of the preferred orientation it matches best to. You see that if we cool, yellow and bluish colors go away, yeah, and we are here in the quantification, we are left with something like an oblique bias. So we come from a cardinal bias to an oblique bias, indicating that actually we have an instruction of this bias, yeah, by the intrinsic connectivity, in this case the interhemispheric connectivity. So Sergio Neuenschwander and Dado, they were asking, yeah, but this was with the cooling, do we see this kind of expectation also in the ongoing activity when we do not manipulate the system? Then we record it in both sides of the visual cortex. And what you see here was the, oh, there's an A missing. It was the thesis of Beatrice Frotta. We measured the coherence between, the coherence of the spiking activity between sites recorded within the hemispheres and across the hemispheres. And what you can see here is that across hemispheres it's quite difficult to get sites which are interconnected. Now, when you implant the electrodes, the coherences are a little bit lower, but they are also higher when the neurons have the same orientation or the same direction preference. So from that, how I am with time. Oh, conclusions and open questions. So let's go to the human stuff. How did we come? So I told you that there are two distinct layouts at least, and we want to represent these simple features like contours and direction selectivity and spatial frequency selectivity. And from that, so what we're interested in to check out with the Göttingen group, and so is there an ancestral layout? Because the South American marsupials hasn't been tested to this question so far, but the Australians showed the pinwheels. So feature selective circuits store perceptual grouping operations. An interesting question would be, do animals without pinwheels perform different operations? And maybe Sigrid has some answers. And I showed you that ongoing activity in callosal circuits anticipates or predicts likely midline crossing shapes or movements. So it could be a horizontal shape or it could be a vertical line which is moving across the vertical meridian. And from that, I asked the question, because I think all the channels, all the talks so far go to this, that it's better that we go to the awake behaving organism and try to get away from the anesthesia. can we establish reliable behavioral markers of the integrity of visual interhemispheric circuits in humans? And there, a couple of years ago, we started to use a very classical paradigm in visual psychophysics. Whoops, does it change? Doesn't want to? Oh. Don't know why. Coming? Now, yeah. So these two dots, this is the so-called motion quartet. Doesn't want to? I think I have another one. Now it's kind of processing things here. So we try to challenge the... I'm too fast for the... Sorry. For the slides. What we try here is actually to... to challenge the interhemispheric connection. So you see this stimulus, which is called the motion quartet, but it deconstructed itself. And it consists of alternating dots, which produce the illusion of an apparent motion. So, it's kind of nasty that it's going away. Yeah, so they switch from here to here, and this creates an illusion of motion, which actually isn't there. And if you present this motion quartet here, right in the middle of the visual field, you use, if you see the horizontal motion from here to here, so it's an ambiguous stimulus. It can actually, if it's presented equidistantly, it can spontaneously flip with 5 minutes, okay, with the different motions. It challenges the interhemispheric connections, and this kind of motion could be processed actually in one hemisphere only. So we tried, so this is a classical stimulus, but we tried to tune the stimulus a little bit from knowing from our data from the cat experiments. So we saw actually that those connections which make the link between the two hemifields, they are not only linking and predicting, but they predict in a selective manner. So we hypothesized that if we pair those elements with collinear or non-collinear contours, we should get a higher likelihood of having horizontal motion versus vertical motion. So in this case, we would predict actually that we have a higher likelihood to perceive a horizontal movement, and here we would have a higher likelihood to perceive movement generated within the same hemisphere, parallel to the vertical meridian of apparent motion, vertical motion. So our first attempt to do this in psychophysics showed actually, and what you see here is actually the likelihood an observer perceives the stimulus as moving like this. When it's more negative, it's more vertical, and when it's more positive, it's perceived more horizontal. And what you see is that actually the only one of those versions of our motion quartet which really made it to a lot of horizontal motion was the one which is paired with the collinear contour, and that apparent motion actually can be tuned subjectively by doing this pairing was already observed in France like 20 years ago, 15 years ago. And in our first version, you see that actually, nevertheless, when we present the motion quartet with the similar distances, the control stimulus is perceived a lot vertically. However, we became a little bit better in this, but it's still significantly different between the vertical and the horizontal. And this is EEG data, which also shows that if there's horizontal motion, we have a little more coherence actually across the hemispheres, which is here seen in the difference picture in the alpha band when with the vertical motion. That was already known. And now we became a little bit better in this, and this is a spoiler alert to the poster of Emmanuel. And then there's one more data slide, and then I'm almost finished. So here we became a little bit better. What you can see is that the control stimulus is actually here kind of a little bit negative, so there's a higher likelihood to perceive it as moving vertically. If we tune it, or if we associate it with a horizontal contour, a collinear contour, we get it a little bit more likely that observers perceive it horizontally, and if we associate it with a horizontal contour, we get it more likely than the control stimulus that it's perceived vertically, so as we actually hypothesized. And I don't show you how aging affects this asymmetry. Will be on the poster. The only thing is that elderly observers actually need a little bit more time to perceive, to indicate that there's a horizontal direction of motion coming on, and this is independently of the three stimuli. This is the first stimulus. And now we also actually try to benchmark this behavioral marker. We do a lot of experiments on testing it, whether it's reproducible in the same person, whether it is also still reproducible after a month or so. So can it actually really tell us something about the interhemispheric network individually about that person? And the idea was not by us. Of course, there are other people who already showed that the interhemispheric transfer ratio really schizophrenic patients says something about the about the velocity the motion quartet is processed. And why should we go to psychotic patients? So is perceptual grouping actually impaired in schizophrenic patients? And that's only one example where collinear facilitation is really impaired in the schizophrenic patients. So the facilitation effect, or you get when you have contours aligned collinearly and you have the middle one with the lower contrast, it gets away when the task gets more difficult. So maybe for the observers which are further back in the audience, this is actually perceivable. And this facilitation you get for if it's vertical versus horizontal, it goes away if it's a patient who has already symptoms or who will later on develop symptoms. And we tested the motion quartet with the three different stimuli in first episode schizophrenic patients, which this is a collaboration developing with the hospital, with the psychiatrists there, Dr. Walter, Dr. Aristella, and Amanda. And Amanda, who is a student of Initiation Scientifica in our lab, did a pilot experiment there, a couple of patients, I think we're 8 patients. And what you see here is that the patients perceive a little bit less horizontal, or the likelihood that they perceive horizontal motion is a little bit lower than perceiving vertical motion. And this is significant, and there's not yet a difference between the stimuli. So we cannot say whether this is something which has to do with the interaction across the corpus callosum or with the effect that they have an impairment in collinear processing. Okay, conclusion and perspectives. Continuous coherence between hemispheres can predict directions of motion. Pairing apparent motion stimuli with collinear orientations alters the perceived direction of motion, and behavioral signatures of stimuli which cross the midline of the visual field in humans may be useful for monitoring the integrity of these very selective visual circuits. Thank you very much for your attention. And no questions for me?
I think we can do the questions in the end of the session, okay? Thank you, Professor Kerstin, the director of Brain Institute, and which composes, together with Professor Claudio and Professor Tarcísio, the organizing committee of this symposium. So now, I would like to invite Professor Claudio Queiroz with the lecture Peephole, using a psychophysics experiment to teach visual perception. So, thank you, Professor.
Okay, good morning, everyone. Thank you, Flavia, for the introduction. And I'm very happy to be part of this session, although most of you know that I don't work with visual neuroscience, but I want to bring a little bit of the work I do as an outreach program that we have in the lab, as a group for outreach to teach neuroscience in schools or in the undergraduate courses. And I want to share with you a new paradigm that's called, I call it the Peephole. In Portuguese, I call it janela indiscreta. That is a way to study how we recognize images. And the reason that I start this program is because this image, I remember the first time I saw this image. So raise your hand who has already seen this image before. So I would say that more than a half of people have ever seen this image, have never seen this image. So everybody saw the dog here? Right, so I remember the first time I saw this image, it was in a FASB meeting. I was a master's student. It was during a talk of a very distinguished researcher, that is Sergio Neuenschwander, and he was very proudly showing his beautiful gamma oscillation in interhemispheric neurons. And I was very impressed. I like it very much, and I thought that visual illusions and visual perception is a very interesting field. And somehow I would say this contribute for me to change my research path. I merged from pharmacology to neurophysiology, so it was a very important talk and a very important moment in my life. This talk of Sergio in the FASB. But of course, Sergio and Kerstin show a lot of results about visual field, visual response or visual cortex in the retina and the LGN. But of course, when we talk about vision, we're not only, we observe the world with your eyes moving around. So we perceive the world not as static, so our eyes are not fixed, but we are moving our eyes all the time. And most of you have already seen these images of Yarbus, that he was one of the first to use eye tracking to record how a human being perceives an image and where this person is looking in a photo, for example. And well, he discovered that this person, or most of people, focus on the eyes and mouth, and even though the face is turned like a silhouette, you can see that the eyes movement focus on the eyes, the mouth, and the face, and that is the reason. One possible explanation is that because we understand the emotions and the feelings or the intentions of other people recognizing the face, right? And later on, Alexander Luria showed that, thank you, showed that if you have a lesion in the frontal cortex, mainly in the frontal eye field region, you cannot move your eyes according to the intention of discovering that image, right? So with all this idea in mind, I start develop or bringing this kind of illusion to schools and to undergraduate courses that I usually give. And I started, for example, making South American adaptation of most of the illusions. So for example, who here has already heard about Margaret Thatcher? Yeah, everybody, right? So, but for my surprise, when I showed the photo of Margaret Thatcher, this Margaret Thatcher illusion, nobody saw it. I didn't find any interesting. And then I just made myself with this girl here. Do you know who is it? It's Anita, right? And the illusion goes that you just change something in the face of Anita, and when the face is upside down, you can see that this image is a little bit different from each other, but if I put in the upright position, the strangeness increased significantly, and I found very, very interesting, and I like to discuss that with the students. And of course, I don't focus on the V1 or the visual cortex as Sergio and Kerstin, but of course in other regions of the brain, mainly in the infratemporal lobe, where you have the face recognition area. And this face recognition area allow us to see faces even though we don't have faces there, right? We can see a face here, we can recognize even emotional states of this non-face stuff, images, right? So one of the questions is, okay, it's nice to show some visual illusions, but is it possible to bring some behavioral paradigm to school so you can teach students and the professors and teachers in school how we see the world and how we recognize the world? So a couple of years ago, myself and Professor Eduardo, we created what we call the Laboratório Aberto. So this talk is from Laboratório Aberto, where we bring cognitive neuroscience and neurophysiology experiments to schools, and we develop experimental paradigms to study that. And we have been, in the last 4 years, more than 20 schools, 3,000 students all over the Rio Grande do Norte, so it's a very nice project. And if any one of the students want to join us and to be part of it, we are open. In the beginning of the year, we start opening positions. So if anyone is interested, you can join us and develop new ideas to how to bring neuroscience to schools and to share information about the brain. So in this type of project, we can give a talk about visual motor integration. We can talk about information processing, how the action potentials in the retina goes along the brain to generate movement. But today what I want to bring is a question: how can we study this visual motor integration during image recognition, right? So we develop a paradigm using PsychoPy. So usually, probably some of you already use this program. So it's a very easy to organize a behavioral paradigm. And you can collect information very precisely from mouse movement or reaction time and everything. So what is the Peephole paradigm? So we start with a hidden image behind the mask. And when the trial starts, this mask, this image is revealed by a small window, so a circular window. And the participant can move this window around. So as the participant moves the window, it can get more information from the hidden image. And when the person or the observer realizes what the image is all about, it presses the start button, and then it can write what's the object it is. And then we can, of course, reconstruct the path and calculate the reaction time that the observer takes to discover or to recognize the image behind. So here is just an example how the track happens, like a GIF. So this is the image that is being, the circle is moved by the observer using the mouse button. So then we can get the data, the X and Y position of the mouse in the screen, and then we can project using these heat maps over the image, and I think it's very nice. So we can, in a way, very cheaply study the eye movement without having an eye tracker. So the eye movement, in a way, is substituted by the mouse movement. So it's a visual motor integration. And we can study how the observer, what's the pattern to discover what the image is behind. So if you look at this pattern, the person starts here, and you can see, as Kerstin has mentioned, the collinearity of the transition between the kiwi inside and the outside of the fruit. And you can see that the person is moving along the sharp contrast. And since it didn't recognize what the object is, it goes inside of the fruit and starts moving along the high contrast between the seeds and the fruit itself. So what are the questions that we can investigate using this paradigm? So how the amount of information and quality of the visual information during perceptual exploration affect the time observers need to recognize an image. So to do that, we manipulate the window size. So we go from a big window, so the person gets more information, to a small window. After Kerstin comment on these results, I calculate the size of the window. So it's 1.5 degree here, and here it reaches 0.3 degrees. So I don't know if it's too big or too small, but you will see the results in a couple of minutes. So the experiment starts with the big size window and progress to a smaller size window. Each session has 5 images leading to 30 images in total. And we also manipulate the color and the category of the image. So we have animals, you have drawings, you have fruits and vegetables, or like architecture or and objects. And of course we can present to a different set of observers the black and white or the grayscale image. The idea is to understand whether color have any meaning in how fast the person can identify the object or not. So what we found is that we can construct this map, so they are really nice. You can see that most of the when there is a face or an animal, for example, the track concentrates in the face, like I showed you before. There is some kind of trophism to the face area. So whenever the person is moving the window along the image, it usually goes to the face and explore a lot of the face. So two different persons and doing the same image, we can see that all of them goes to the face in the end. Even in the drawings, we can see the face. And we can see that the Gestalt principle that usually map the object along the figure background transition, right? So what we observe is that heat map shows the exploration pattern and focus on higher contrast and perhaps high spatial frequency, so there is more information in it, and usually goes around the faces. So, for example, how the window size affect this exploration pattern. So here we can see different observers, and we can see that the high window size, the observer moves not a lot, the window. As the window size decreases, the amount of movement, and therefore the trajectories increases, the path, and the reaction time as well. Okay, I'm just finishing. It's very fast. I'm just give you an idea. So here's the quantification. We have like 14, 15 people per condition. It's not the same. So the person that goes to the color condition do not go to the black and white or grayscale condition. And what we found is that as the window size decreases, the reaction time increases. There is a clear increase in the reaction time for grayscale images compared to the colorful ones. And of course, the correct response, so the accuracy to identify correctly the object, decreased very much at window size 6, that is 0.3 degrees. We found that if the window size is smaller than that, it's impossible for the person to recognize what the image is behind the mask. When we compare the reaction time across semantic categories, for our surprise, we didn't see differences for objects, animals, architecture, or drawings a little bit, but those categories that shows the highest difference between color and grayscale images is the fruits, and perhaps the reason for that is that the texture brings a lot of information, so the fruit itself, when it's black and white, you cannot use to determine what is a pear or an apple, for example. So therefore, if this grayscale or black and white image, I put it here a color, but it's the path that you see here is a grayscale. You see that the person has to scan the object a lot more to discover what the object is, what the image is. So as a conclusion, I believe we were able to demonstrate that the Peephole paradigm proved to be a viable and sensitive method for detecting patterns in the observer's visual exploration behavior. So you only need a computer, a laptop, and you can do the experiment in schools. Also, the heat maps reveal perceptual strategies consistent with the Gestalt principle, so we can discuss Gestalt principle using this paradigm. Window size strongly determines recognition time, so the smaller the window, the person needs more time to detect the image, and of course, color plays a significant role in the recognition process, and mainly for objects or images that color plays a key role in discriminating between them. This work was initially, the first version of the software was designed by Wigna Farias, who was a recipient of a scholarship in the lab 2 years ago. The first behavioral paradigm, the first experiments that we performed in school was done by Bruna, Eduardo, and myself. And this year we get the join of Gabriela, Hayuri, and also Ana Augusta, that is always present in the Laboratório Aberto, and Ana Clara, who recently defended her TCC using this paradigm as a part of the Laboratório Aberto team. So thank you very much for your time and attention.
So we have some minutes to questions.
Just a short curiosity. Doesn't the initial position, the the initial peephole influences the reaction time as well? I mean, depending, you can start on a face or far away from it?
Yeah, usually we try to, the initial position is always in the center of the image or around the center, because there is a random positionness, but it's never on the face, so the image is placed on the screen that the face is not in at the center.
So the initial position you guys set is not something the participant...
No, it's set, but since the mouse is getting the information all the time, if the person enters the session and already moved the mouse, the initial position can go to a blank space. That's something that we didn't want. We want the initial position to be inside the image, so the participant can see where the circle is, because I don't know if it was in the image I showed, there is a black line around the circle, but in the paradigm there is no line. So the initial position usually is in the center of the image, far away from the face.
So we have time for the questions for the Professor Sergio and also Professor Kerstin.
Please.
Because you said that the mouse positioning could be a surrogate, or could be like moving the eyes.
Well, it could be a
This
way of how we explore the image without the need of an eye tracker. But of course, the eye movement is much faster, and it has a corollary response.
The fact that is fast and effortless changes the game. For instance, the description of this Lévy series in exploring the images will not appear here, just because you cannot move the mouse like putting the mouse
Very fast, yeah.
Yeah. So this is a big difference. Also, it would be nice if you have false colors, for instance, green bananas or violet bananas,
Yeah.
instead of just comparing the color. Yeah. But I find it interesting and
That's why I put myself, since I'm on the organizing committee, I was able to put myself in this session. So it was on purpose just to know your opinion about it, and Kerstin opinion, secret, of course.
There are many things to think about this.
No, we have many ideas. So we want to scramble the image so you can get an information of a patch. But since you scramble the entire image, you violate the continuity and the. Yes, I agree. Epilepsy field, yes, probably. Who knows?
I had kind of a related question about the comparison between the eye trajectory and the mouse trajectory.
Yes.
I feel like for a couple of other reasons you might expect them to be different, because when we are looking around an image, we are making saccades with our eyes. It is known that the information that we are using to decide how to explore depends on what we are seeing in the periphery. So we are making a fixation, but it's not like we are only seeing the stuff in the fixation and then everything else is blank. We just kind of see like a lower resolution, but we are using information from that lower resolution stuff to make the next saccade. But when you have this mouse thing, you basically have the information make a step drop to 0. And so you are not getting the peripheral information which you would use to make the next jump. So I feel like for these reasons, I would not expect this to be a very faithful proxy of how the eye movements would be. But it would be interesting to compare the two and see what are the things that are different between a natural eye movement exploration and this mouse movement stuff.
Yeah. Thank you, Daz. Indeed, it's very difficult to, first, I mean, eye tracker experiments are very complicated and expensive, and very difficult to bring this setup to schools or to a classroom like this. So I agree with you. So in this paradigm, we remove the peripheral vision. So we do not use the peripheral vision to integrate into our, let's say, peephole. And also we make a lot of demands on our working memory, because you must remember what you just saw and you must integrate without looking at it. So I agree, there is some important difference, but discussing with Kerstin, I was just wondering whether we can apply this type of paradigm to let's say, people with dementia to see whether the reaction time will increase because the memory load or the working memory load is too much for them to keep track of what they have just seen. And maybe you can reveal something about the brain or the behavior that can be relevant for clinical settings.
But I think the suggestion here is exactly the other way around. You could consider to put blurry, and you can put very strong blurry, even compare with the magnification factors. So you could put the logarithm
Decay.
blurry. And then now you could compare the other movements and the output. And then you could say what you see.
Well, if I get to put my hands on an eye tracker, maybe we can do this experiment and compare how it goes. I see. I see. Maybe, yeah, well, I see. Yeah, nice idea. Stronger or yeah. 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 Like a phase lower, fade out of the previous spatial exploration area. Yeah.
Very interesting data. I want to ask if anybody have questions for Professor Kerstin and Professor Sergio.
I have a question to Kerstin.
I'm sorry, your question is about, yeah? Thank you.
Kerstin, do you have a question to me?
Anyone? No? So,
Okay, I have a question for Sergio and Kerstin, but first I want to have one more question here. So you've brought this to thousands of students by now, or something similar. So what I'm wondering, you're teaching both vision and the logic of science.
Yes.
So are there in these classes then kind of schools of thought that have different models of how their visual systems work, and then they find the truth by looking at the data? Or so it would be interesting to have a few experience reports. What does it make? Is this a way to get students think more in the logic of science, which is super important, of course?
Yeah, thank you for the question and comment. In fact, the goal of the Laboratório Aberto is to foster scientific inquiry in schools. So the idea is to discuss scientific methods. And so far we have collected more than 20 experiments that we can do at schools. And I invited all of you to look at the Gabriele and Hayuri poster in the poster sessions that they will describe muscular contraction, right? There is data from muscle activity and fatigue, for example, to study how you can study fatigue at school and to understand what fatigue means from terms of electrophysiological recording, EMG. So we have many experiments, and these experiments were not given to the 3,000 students, but usually we have a talk. We go to the school first. We give a talk to the entire school community. Usually it's like 300 people in one talk. And then after that, we have the workshop where we invite the professors, the teachers from that school that goes to one of the workshop, for example, visual perception. Then we discuss what visual perception is. We bring some information, content, and then we bring the experiment. Then they do the experiments by themselves. We analyze the data, we discuss, and then we close with a discussion with the group. And then our expectation is that the teachers from those schools will bring these experiments to their students in the school, right? So that's the idea.
Yeah, thank you. So that's a question for Kerstin. So with this, your kind of predictive coding interpretation of stimulus moving from the cooled hemifield to the intact hemifield or the other way around, I would like to understand a bit more what the prediction does, what the accumulation of evidence does.
So
maybe I got it wrong, but is it that the effective field is bigger if the stimulus comes from the cooled side, or yeah?
Yes. the effective field is bigger, and the spontaneous activity is lower if that part is missing. So it's kind of apparently raising the activity of those neurons, or they are spontaneously, the activity would be higher, and that part of that connection which is missing is contributing some ongoing spikes. That's actually an indirect prediction.
So I want to thank you. We have to close the session because the advance of time. So we come back 2.5. That's it? Yeah. Thank you very much for inviting me, and see you.